Hi, everyone. Welcome to another Science on the Sound here with the Mass Center Aquarium. We are super excited and kind of sad because our speaker for today is Emily Bjornsgard, who is our current lead aquarist. But she will be leaving us to pursue a degree at Western and so we are. This is like your last talk with us, Emily. I'm so sad. If you haven't seen all of her other great talks that she's done over the last 2 years, they are on our Science on the Sound speaker series tab, our playlist. She's got some great ones about Pacific spiny lump suckers, which are her favorite fish. She's got one about jellies. So you'll be missed, but we're super excited that you've decided to do one more talk with us before you leave. And this one is all about the seasonality of Poverty Bay, which, if you don't know, that is where the Mass Center Aquarium is stationed. Poverty Bay is the body of water, and we're kind of smack dab in the middle of Poverty Bay. So, without further ado, Emily, take it away.
Awesome. Thanks, Ariel. Like Ariel said, I'll be talking about the seasonality of Poverty Bay, especially what's kind of been going on in the last 2+ years that I've been here at the Mass Center. And so let's get right into it. Uh, so a little bit about me. Ariel had a great introduction. I am the current lead aquarist at the Mass Center, which means I take care of all of the animals. We have about 250 species in about 3,000 gallons of water. So it's a lot of diversity in a very small little package. But I am the lead aquarist until September 10th. This Wednesday will be my last day, and I am moving up to Bellingham, Washington to pursue a master's degree at the Western Washington University, which is based in Bellingham and has the Marine Lab in Anacortes. I'm super excited to get up there and start my program. I'll be working with the early life history of forage fish, specifically surf smelt and Pacific herring. This picture here in the middle are some surf smelt embryo in eggs. I got to go up and visit about a month ago, and I'm super excited to get started. And as you'll see in this talk, I am a big fan of all things baby marine life. We see a lot of it here at the Mass Center and in the greater Redondo Beach, Poverty Bay area. So I'm super excited about that. I am very, very sad to be leaving the Mass Center, though. It's been an incredible time here so far. In addition to all of that, I am also a scuba diver and cold water snorkeler. So I've spent a lot of time both on the beach and in the nearshore kind of habitats, both at the surface and under the surface. And I also am a marine life photographer and intertidal or beach explorer. You can find me on Facebook and Instagram at the handle eMarineLife. These are just a few pictures of some of my favorite moments. So we're going to be talking about Poverty Bay and specifically the Redondo Beach dive site, as well as the beach itself, and then what is happening both in nature and in the Mass Center Aquarium. This map is a dive map of the main part of the dive site here at Redondo Beach. And then divers, if you're a diver and you're watching and you've ever dove Redondo, there is a good chance you've seen this cable that comes off the end of our pier here. That is the cable down to the Mass Center's pump. So the Mass Center pumps in water 24/7 into our aquarium exhibits, which then drain back into the bay. So even though it is an aquarium and they are a kind of closed tanks, they are an open aquarium system with the flow constantly coming in and constantly going out. And so we get a lot of the same environmental cues and ecological events that happen in the sound happen in the aquarium as well, which makes it really exciting. There's always something new happening, and it also gives us a chance to kind of study what's going on in the bay in a more controlled, semi-controlled exhibit such as the aquarium. These are a few pictures of what we have as far as marine life in the aquarium. Our Up until this last summer, our largest animal was about 6 feet long. Her name was CQ, the wolf eel. She has since retired to Point Defiance in a much larger exhibit, which we are very excited about. And some of our smallest animals may only be a couple millimeters long when we have plankton that comes in and settles in the exhibits. Um, And then here is a picture of some of the masked aquarium divers doing a skills refresher dive, just to make sure we're up on our skills. So we think about seasons, of course, spring, summer, fall, winter. The seasons themselves might be different experiences in different areas, but we all know the four seasons. Specifically here at Redondo, though, we can track kind of what season it is based on the visibility in the water, what animals we're starting to see in the nearshore ecosystem, when the lowest of the low tides are happening, whether that's in the middle of the day or the middle of the night, because all of that changes throughout the year. These are just a few of the events that kind of seem to be standardized throughout the year. Everything shifts a little bit each year depending on weather and other kind of environmental cues. But these are some of the things that we have seen on a regular basis, and a few of them we will go into more as we go through this talk. And so where did these observations come from? This is not a like peer-reviewed science presentation. This is something that we have observed, I specifically have observed over the last 2.5 years, and that have been observations from other staff members and other members of the community throughout the past couple decades. Some of this is from our collection opportunities that we do for the aquarium. Here at the bottom, this is a beach seine net, which is about 5.5 feet tall and 50 feet wide. We can bring it out in the shallow water at low tide so that we can get about 6 to 10 feet below the average low tide line. And by collecting these animals, most of them go right back into the bay. We're looking to see what animals are utilizing the nearshore habitat, what size of animals and whether they're juveniles or adults might be found there, and also what the habitat itself is looking like. Is there more eelgrass? Is it more rocky? All of these things kind of help inform what the bay is experiencing, and helps us see what the bay is experiencing. And so we also, during these trips, if there's a specific animal we would like to add to our aquarium collection, we have all the permits through Washington Department of Fish and Wildlife to do so. However, most of this is just looking and analyzing kind of our local habitats. We will also do beach walks. This picture here is low tide at the south end of Redondo. There's a lot of eelgrass and kind of sandy flats, which is different than a little bit further north where it's mostly rocky and has a little bit more kelp. This is the picture of the aquarium itself. As you can see, the water draining from the building. That's normal. That means that our pump is working, that water is coming into the building and then draining out. And then this is a look off the end of the dock into the dive site from the surface. So all of these different areas inform kind of this presentation and what we've been seeing in the seasonality and the changes throughout the year here at Poverty Bay. And another way to look at the seasons is to look at the plankton. So I personally have a background in phytoplankton monitoring as a volunteer through both the Olympic Regional Harmful Algal Bloom, or ORHAB, on the outer coast when I was a little bit younger. And then now we partner here at MAST, we partner with Sound Toxins, the kind of inland Salish Sea monitoring organization for any harmful algal blooms or just algae blooms in general. And so we see some trends through the plankton as well. So we're going to look kind of at both the microscopic life that is inhabiting the Poverty Bay ecosystem, as well as kind of the macroscopic, or the bigger things that we can actually see with our eyes. So for those who don't know, plankton is anything that drifts. The word plankton comes from Greek planktos. And this could be the microscopic things that you may just see a haze in the water, or if something's blooming, you might see just a solid color in the surface waters. Those are going to be our microscopic plankton. So our diatoms, or our plant-like plankton that photosynthesize and use carbon dioxide and produce oxygen. That's these two of our many diatom species are shown here. Dinoflagellates can also photosynthesize in certain species, but they can also be predators. So they can either be autotrophs, meaning they're photosynthesizing and producing energy and oxygen, or they can be heterotrophs, meaning they are eating other organisms for their food. And dinoflagellates are single-celled right here. These are two of the ones we see here. And then zooplankton are often, but not always, larval or baby forms of some of the larger animals that we see on the beach. So in this two photos here, we have some barnacle nauplii or baby barnacles, which tend to pop up right in the late spring, early summer. And then you start to see them on all of the rocks in the intertidal zone. And then this is a crabzoea, so the first kind of freshly hatched stage in a crab's life cycle. Jellies are also considered plankton because they drift. They don't have any really true directional movement against currents. They may go in one direction, but they don't fight the currents, or they just kind of get drifted where they're taken. So here we have a red sausage jelly, a moon jelly, a comb jelly, and an egg yolk jelly. And based on what is blooming and what is growing really quickly in the plankton, the bay may look very different. These three pictures were taken this last summer. Earlier this year, we had Chaetoceros debilis. It's a chain-forming diatom, so they're the plant-like ones that photosynthesize. They are not toxic to humans, though they do have spines that in super high amounts can cause damage to the gill tissue of certain marine life. This one did not seem to cause any issues, but it was very pretty under the microscope. As you can see, it had kind of a brownish, beigeish color to the water when it was concentrated. And so that looks a lot different than the Noctiluca scintillans bloom. This middle one, there was so much Noctiluca. This is also called the nightlight dinoflagellate. They do eat other organisms, often other like diatoms and smaller cells that they can engulf with the eyeball-like cell. And as you can see, when in really calm water, Noctiluca sits right at that surface. And when there are so many of them in the water, they can actually block out the sunlight needed for phytoplankton that live deeper in the water. And so they are really good at taking up that resource and becoming kind of the only thing in the bay. And then Thalassiosira and others, this is a mix of a few different diatoms. So again, plant-like species. And as you can see, because the cells are so small, if you have a really big bloom, it may turn the water really, really dark, almost opaque. But it also depends on how many per a certain volume of water. So this was just a scoop of the surface water. You can see it's fairly clear. This beaker here was the result of a plankton tow net. So we towed a net through the water and concentrated that sample down. So the surface water didn't quite look brown like this, but it did have a bit of a brown tinge to it. So starting with the fall, because that's where we are in the fall, almost. I'm gonna just claim that we are in fall season. During September, we start to see a transition from summer conditions into kind of colder air, colder water. The visibility in the surface waters and in the kind of diveable depths tends to get better. There are always those days where it's super hot and sunny and the plankton just blooms overnight, and then the visibility just gets really, really bad. But generally, it's starting to get better because those phytoplankton that are in the water are starting to die off. There's less and less nutrients because they have all been used up that summer. And so generally, if you're diving or if you're snorkeling or if you're kind of using the nearshore habitat, you're starting to see better and better visibility. These pictures were all taken between September and November in the last 2 years. So there's definitely still some really cool plankton in the water, but we're starting to see kind of a decrease as we get closer into the fall. As far as larger animals in the nearshore kind of habitats, we start to see opalescent or market squid, which we'll talk a little bit about specifically. Sometimes we see nudibranchs kind of towards the end of the summer laying eggs, especially on the beaches. And then one thing we'll talk about in this section, because it started in November, was in 2023, 2024, we actually had our resident grunt sculpin, a really cute little fish here at the Mass Center, lay eggs. And then that turned into a year-long project learning about their larval development. So it was super, super exciting. So looking at opalescent squid, you might see these clumps of squid eggs on the beach at any time. Squid are always kind of in the process of laying these eggs, depending on where you are. But we tend to see these a lot during the late summer, early fall, where the adult squid are starting to move through the sound. There is also a fishery for squid, so recreational fishers will go out and catch them. In the past, Mass has celebrated Squidorama in November, which is kind of the celebration of the squid moving through the sound. And so one thing we've done here at the Mass Center is look at these squid eggs and try to see the development of the embryonic and early larval history of these squid as they grow. We have these photos here. This is from a couple different trial runs with a couple different egg capsules. Each capsule can have over 50 eggs per single sack. And if you look back at this picture, there's several dozen egg sacs. So there are probably 1,000 eggs in that cluster right there. This first picture is a freshly laid egg sac. You can see those eggs have no real distinction between a yolk forming and the beginning of a baby squid. As you move kind of from left to right and top to bottom, you can see that the body of the squid starts to form on one side, and then the yolk sac forms on the other. This growing baby squid it kind of absorbs the yolk sac. You start to see the eyes develop. Squid have very complex eyes as far as invertebrates go. You can start to see their color changing cells show up on the body. There's some tentacle growth around the mouth. And then you can start to even see their inner organs start to grow. Over 3 to 5 weeks, you end up with lots of little baby squid. Another species that we start to see a lot during kind of end of August to kind of mid-fall is the hooded nudibranch. They, again, can be seen year-round, but in this area, historically, this is when we start to see them more often in kind of the intertidal and really shallow subtidal zones. Hooded nudibranchs are a great display species for aquariums, and we've really enjoyed having them here at the Mass Center because they don't eat sponge or other encrusting organisms that you also have to grow. They will live very well on brine shrimp, which is a common live foods thing that we can grow here at the aquarium, and we already were growing for our jellies. So we were able to keep these hooded nudibranchs alive for over a year, which if you know sea slugs, they have notoriously short lifespans, so that was a very exciting thing. This video here kind of shows them using their hood to scoop along the side of the exhibit. They also will eat plankton out of the water column. So this picture down here, you can see this hood is open, you can see that hood is open, and they will catch all those little dots, which are the little brine shrimp. And also in this picture, you can see some eggs. This summer and early fall, our hooded nudibranchs were laying eggs like crazy. So they are something that you can find in eelgrass beds, primarily, so kind of at the south end of Redondo, south of us to Dumas Bay, kind of Dash Point, and north of us to Burien, Seahurst area. The Gruntsculpin, this was a project that I haven't done a full like science on the sound talk on, but I definitely wanted to touch on today because it was one of my favorite projects that I've worked on here at MAST. Gruntsculpin, scientific name Ramphicotus richardsonii, are just bizarrely adorable fish. They are bottom fish. They, as you can see, they live on the bottom. They kind of hop around. They can swim. They choose not to most of the time. Their face looks like a closed acorn barnacle. Their tail looks like a feeding acorn barnacle. They often live in barnacle structures. And they can live a long time, up to about 13 years, some sources say. These two shown in this video were our two resident Gruntsculpin in late 2023. And up until that time, we had gotten eggs, but they never really hatched, according to past staff members. And so we were kind of hoping for eggs, but we didn't have really high hopes. But we didn't even know if they were male and female. Obviously one of them was female because there had been eggs in the past, but there wasn't any confirmation on which one was which, or whether it was even possible to for them to have fertilized eggs. So We did start to notice this left picture was taken during the summer, about July, I believe. And then these photos were throughout October and early November. You can see kind of a rounding of her belly, and these were on days that she wasn't getting fed. So either she was eating something in the exhibit that we didn't know about, or she was going to start laying eggs, which was super exciting. So we kind of watched her, gave her her space, and after probably 2 weeks after that really round picture, we saw both of the grunt sculpin in one of the barnacles, which was abnormal. Usually it's one at a time. And then the next day we came in and there was a great little pile of white, kind of rice-looking eggs that the male grunt sculpin was guarding. Again, at that point, we didn't know that there was a male. We just knew that this was the non-female at that point, because this is the one that did not get bulbous. But he was very proud. Grunt Sculpin will guard their eggs, so that is a big deal. And so we watched for 3, 4, maybe 5 weeks, and didn't see a huge change, but you can see that those eggs have a really thick kind of shell to them. They're opaque, unlike a lot of fish eggs that we see, where you can actually see the development inside pretty easily. And so after about 5 weeks, we couldn't really see anything in the egg, or in the barnacle like this. And so we figured either they aren't fertilized and he's just guarding them because that's what they do, or they are fertilized, and we're going to take a closer look at them to make sure that there aren't any rotting ones or anything. And so during a cleaning, we pulled them out of the barnacle as gently as we could. And so we did that. And as you can see in this middle photo, once we were able to get a stronger light on them, that there are actually eye spots starting to form in these eggs. This was about 5 weeks post hatch or post spawning. And then in mid-January, we saw something really exciting. We actually captured the heartbeat in one of the eggs, that you can see through this thinning spot kind of on the shell. So that was a great sign that we knew where we were going. We were heading in the right direction. And so then early to mid-February, we had all of the hatches. Over 90% of the eggs hatched. This picture is a fish in process of hatching. Doesn't look super thrilled to get its picture taken, but it did great. It hatched just fine. And as you can see here, grunt sculpin don't look like grunt sculpin when they hatch. They have very, very tiny and transparent tail fins or caudal fins, and very minimal pectoral fins or their side fins. And they are planktonic, which means they're in the water column. They're not necessarily on the bottom hopping around. They are true swimmers when they are babies. So that was a whole nother level of trying to figure out how to best keep them so that they are getting all the food that they need, all the nutrients that they need, and all of the kind of cues that they need to go from a swimming planktonic species individual to a benthic bottom individual, which was something we did over a period of a couple months. It took about 3 months for them to transform from their larval form to their benthic or bottom-dwelling juvenile form. As you can see, the difference between March and April in size is not very much, but the development is quite a bit. You can see the tail, anal, and dorsal fins starting to form. And the difference between April and May is both quite a bit in terms of length and in terms of starting to look like a real grunt sculpin. Between May and June, we saw that solid transition from planktonic or in the water column to benthic on the bottom. And then throughout June to October, we started to see that pattern start to really show up. They got a little bit more vibrant, and they truly did look like they should, just very, very tiny and maybe even cuter than the adults, because who doesn't love a teeny tiny fish? If you would like to learn more about the grunt sculpin and our experiences doing that, you can search Drumming Croaker. It's an online Aquarius journal. I wrote up a 9-page paper on the experience, and you can learn a little bit more about the grunt sculpin and our experience here at the Math Center. It was really, really exciting to see this transition from gravid or pregnant female grunt sculpin adult to all of the babies, and kind of our process through that. So moving on into winter, starting with the kind of non-living characteristics of winter. Obviously, it's very cold most of the time. It's cold air, but it's also very cold water. So if you are a diver and you're doing multiple dives in a day, you want to make sure that you don't get too cold in between the dives because your surface interval is really cold because you're wet and then the air is cold. There is, however, really great diving in the wintertime because there is limited plankton in the water, which means your visibility and you're able to see a lot farther. Um, Here in Redondo this last winter, we saw up to 50-foot visibility, which if you're in the tropics, that sounds like a really bad day. Here in Redondo, on a good day in the summer, 10 feet visibility is great. So in the winter, when you can see the surface from 50 feet down, you're really enjoying that really cold dive. Some of the animals that we start to see in the winter are roughback sculpin start to lay eggs and lay eggs really constantly. So we got to see that. And also in the near shore and dive site, Redondo kind of becomes lumpy land, which we will talk about as well. And then in 2024, we actually were able to collect a couple lump suckers and then raise babies. So that will be talked about kind of in this section. So roughback sculpin are a mid-sized sculpin with a really tall dorsal spine. I unfortunately forgot to put a picture on this slide, but these are what roughback sculpin eggs look like. They are pinkish and they're usually laid kind of in algae like this, so they're kind of tucked away. This picture here is a kind of early in development photo. You can start to see kind of the shape of the fish in a couple of these eggs, but not much more. It took about a month, 3 to 4 weeks, for the roughback sculpin embryo to develop from this to just before hatching. And then when they hatch, they are only about 3 millimeters long. In comparison, the grunt sculpin, when they hatched, were about 6 to 7 millimeters long. So these are very tiny fish. We did not attempt to raise them just because we don't have the infrastructure to do that. However, because most of these eggs developed and hatched on exhibit and we are an open system, those larvae will just flow right out of the tanks. Whether we want them to or not. So they can go out into the greater plankton um, uh, community in the surface of Redondo Bay. Or Redondo Beach and Poverty Bay. This video, though, is pretty exciting. You can see the heartbeat as well here, just like the grunt sculpin. And then here, these two dots kind of floating in nothing. Those are the otoliths, or the ear bones, of this developing fish, which eventually can tell you how old a fish is if you are able to extract and look at that. And then talking about Redondo becoming lumpy land, Pacific spiny lumpsuckers, as shown up here in the corner, are my favorite fish, as Ariel mentioned in the beginning. They typically reach lengths of about 2 inches in this area. There is a report of one reaching 5 inches somewhere. I would love to see a 5 inch long Pacific spiny lumpsucker. But they are fairly small. They look a little bit like a bumblebee when they swim. They are capable of swimming. They are not very streamlined swimmers, but they live from about Oregon, Washington, and north. They prefer very cold waters. And while you can find them year round here, especially in different life stages, the best time to see them is when they start to come shallow in the wintertime. This video was taken by my dive buddy in January 2023, so just before I started working here at the Mass Center. And we were diving just south of the Mass Center in what is known as Lumpy Land. And normally I would not, I don't necessarily say like go touch the animals, but in this case this fish touched me. As you can see, there was a little bit of a current that day, so I wanted to just like keep my hands stable and not let the little fish fly off into the unknown. But this was one of the most exciting experiences I've ever had diving, was just the little lump sucker landing on my finger. I felt like the chosen one. But fast forward to winter 2024, And the Mass Center was able to collect 5 Pacific spiny lumpsuckers, 4 females and 1 male. This is Papa Lumpy, aka Rusty. He was named that for his bright orange coloration. And we knew that he was a male. Males are usually more circular rather than oval, quite a bit smaller with that really tall, upright dorsal fin here. And so we knew this was a male, and we also knew that he was a male because he started nesting immediately. He found the smallest barnacle cluster or barnacle opening in his exhibit and started making it ready for eggs, which is what the male lumpsuckers do. The females are more pelagic. They will lay their eggs and then continue on their way, whereas the males will guard the eggs until they hatch. And so that's pretty much what happened. This is again Rusty in his small barnacle getting ready to accept some eggs from one of the females. These are the 4 females in a time-lapse video. All 4 of them were gravid or holding eggs, and so we were waiting to see which one would lay their eggs first. We didn't ever confirm which one was the mother of the clutch of eggs that was guarded, but all 4 of them ended up laying their eggs over about 72 hours. And so it was great. None of them had any trouble with laying eggs, which was very exciting. Sometimes they can not lay the eggs and then get egg bound inside. So they all laid eggs. Some of them were guarded by Rusty, and over about 6 weeks, we saw those eggs start to develop. One egg was knocked out of the nest fairly early. That's how we got this picture, and this picture, which was disappointing that he wasn't able to care for all of them, but it was exciting because we were able to see that development in real time without disturbing his nest. And that egg did end up hatching on time with all the rest of them. So no harm done. You can see, though, that staff care of an egg and Papa Lumpy care are 2 different things. With these eggs, even though we were keeping that water that the egg was in as clean as possible, it did start to grow diatoms and other kind of microscopic organisms on the outside, whereas this egg that was cared for by Papa Lumpy up until about 2 days before hatching is crystal clean. So these father lumpsuckers really take their duty as caretaker of the eggs very seriously. And then once the lumpsucker babies hatch out, they have a fully functioning suction cup. So they are able to stick to things pretty much right away. And they are about 5 millimeters long from the tip of their nose to the tip of their tail. However, most of that length is tail. So their actual body is only about 2 millimeters long, which is very tiny. This video here is them on a like 1/4 inch wide airline. So so tiny, so cute. And we ended up with a lot of them. We were anticipating about 200. We ended up with almost double that. This video is taken during a water change, so they're a little bit more condensed than they would have been normally. But as you can see, all of those little black spots with tails are the baby lumpsuckers. And we watched them develop. They do hatch fairly developed. They have the beginnings of all of their fins. However, those fins start to fill out, and their body also starts to fill out as they grow. They got a steady diet of brine shrimp, as you can see in this video here. And those brine shrimp were enriched with a variety of supplements just to make sure that they were getting all the nutrients that they need. And in this photo, you can see the beginnings of some of their body armor or their enamel-covered tubercles right here on the side of this little baby lumpsucker. And over the course of about 8 months, we were able to grow up enough to send to several of the other aquariums in Oregon and Washington. And as of, I think, about last month, Point Defiance Zoo and Aquarium has several of these babies on exhibit in one of their Pacific Seas aquarium exhibits, which is super exciting. So moving on to spring. Spring is when we have the coldest water because that water has been getting colder and colder and colder throughout the winter. But we also have warming air temperatures, which means that there is a lot of nutrients in that cold water for the plankton, and we're starting to get sunlight. So it is the perfect recipe for an explosion of microscopic plankton in the water, and for our macroscopic algae on the beach. So our sea lettuce and our kelps and everything like that starts to grow in the springtime, and grows really fast. And for the microscopic plankton, that means that that water is really hard to see through. But even though it looks a little scary, it is really good for the ecosystem to have this bloom. It's not a bad thing. If you see color in the water, it can be a harmful algal bloom, but it oftentimes isn't. Many of our harmful species actually don't produce a color in the water at levels that we're concerned about. So organizations like ORHAB and Sound Toxins are constantly monitoring the water to see what's blooming and whether it could be a cause for concern. So in the ecosystem, we're seeing this huge phytoplankton bloom with all different types of species, kind of fighting for that top spot in terms of quantity. We also saw our moon jelly polyps start to produce baby jellies here in the Mass Center, and we also had several of our snail species as well as other species laying eggs, having those eggs hatch on exhibit, and all sorts of exciting things. So this slide, we won't go too deep into phytoplankton. If you want to hear more about phytoplankton, check out the Science on the Sound YouTube playlist, because I did a whole talk on phytoplankton. I'm a big fan. But these photos just kind of demonstrate the huge diversity that we see in the plankton during the springtime. I have this picture here again where you can see the unconcentrated water and the concentrated water. And that just shows you kind of, even if you can't see it before it's concentrated, there is so much life in the water that is sustaining kind of the whole ecosystem from the bottom up. Some of these plankton species will chain, so that's like this one here and this one. Those are both Catossaurus species that will form chains of really small cells, and sometimes even form these clusters of cells depending on the species. And then these large species that are called Cossinodiscus sometimes can get even big enough that you can see them with the naked eye. That's oftentimes in the later spring and early summer. And then some of these other dinoflagellate species are really kind of bizarre, and I think they're really cool. This one I like to call the honeycomb dinoflagellate. It's called Polycracos because the central flagella, or kind of whip-like tail that moves it and makes it spin, wraps around multiple times. And so that's a really kind of interesting thing. These are Noctiluca, which we will have a whole slide on later. And then these two species are two separate species, but they have something in common, that is an eye spot in a single-celled organism. So in this one, it's this red and white kind of structure here. And then in this one, it's the white and kind of blackish structure here. But that's called an ocelloid. It's part of the single-celled organism. Again, one cell, but the ocelloid acts like an eye. And so they're a single-celled organism that has an eye-like thing, which I just find fascinating. They are both heterotrophic, which means they are hunting other phytoplankton for to eat. And then this one has this long, straight piston off the back. And I could never find why it has the piston, but it does move back and forth, whether it's for movement or for predation and hunting. I'm not sure, but it's just so many little structures that make up this incredible single-celled organism. And then moving on to moon jellies, which again, also plankton, but they're kind of macroplankton, a little bit bigger. We can see them. Moon jellies have a two-part life cycle, just like all true jellies. They have their medusa, or their quote-unquote adult stage, where we can see them floating in the water. Typically in the late summer is when we start to see them here in Poverty Bay. And then they have their polyp stage, which is down here at the bottom. And the way these two are connected is the adults will spawn. So the females will hold on to her eggs. The males will release sperm into the water column, and then the females will release fertilized embryos known as planula. That's here on the bottom left. And then these planula will find a solid structure to attach to and develop into polyps, which are these here. And then also this is a big clump of polyps. They look a little bit like a long stringy anemone because they are related to anemones. And then each of these polyps can undergo a process called strobilation, where the polyp stretches out, those tentacles kind of get sucked back in, and it forms these flat structures called strobila, which then further develop into these flower-like sections that pop off as ephyra. There's a couple in this photo here. As you can see, they are very small, just a few millimeters across. And then these ephyra will eat a variety of plankton and then eventually grow to form more medusas. And these polyps can strobilate multiple times over multiple years. And so we saw this in the kind of early spring this last year. And the moon jellies on exhibit here at the Mass Center, if you come visit us, are from these polyps that strobilated naturally due to environmental cues in the water coming in. And then these pictures are kind of a side note, but I think they're really neat. Oregon tritons are a species of large sea snail that we have on exhibit here at the Mass Center. They respond to cues in the ecosystem and will lay their eggs without any prompting from us. This picture on the left was taken about 6 weeks before this picture on the right. And it's the same cluster of eggs in all the pictures. So you can see that this snail stays with its eggs, and it also is just continuously laying them as those earlier laid eggs in the middle start to hatch out. The larvae from these eggs will join the water or join the plankton community in the water. And again, they're so small and microscopic that they will just flow out into the greater Poverty Bay. And then finally, our last season to talk about here is June through August, or summer, which we just left. And so we've got warm water, and I put warm in quotation marks because I think it's warm. It's definitely warmer than our winter, but it's still probably objectively cold. But we also have warm or hot air temperatures, which can lead to big phytoplankton blooms, but it can also limit the amount of nutrients in the water. So all of these factors are kind of fighting with the plankton to see which species are capable of blooming really quickly, and then that bloom will die off, and another plankton species will bloom again. So we get this bloom and bust phytoplankton, which leads to variable water visibility and water clarity. We also see our extreme low tides during the middle of the day. During the winter, we see them at the middle of the night here in Poverty Bay. In the summertime, it's the best time to go on the beach and not be freezing cold. So you can go and it's warm and the water is receded way back. You can see all of the cool animals on the beach. So it's a great time to explore the shore more comfortably. So our Noctiluca scintillans, I've talked about this species a little bit. It is a dinoflagellate species, and it is one that I think can alarm people the most because when it does bloom, it turns the water this like radioactive orange tomato soup color, because all of those cells reproduce really quickly, and they also sit right at the surface of the water. So you can see this beaker here, I let it settle, and all of those cells packed into the top like quarter-inch of the water. And so it can be alarming. This species is not considered harmful in our areas. In other areas of the world, it is thought to be a harmful algal bloom because when you have so many cells and they die off, it can produce a lot of ammonia. But here we do not consider them a harmful species. And so if you see this, it's really neat, but you don't have to be concerned that there's an orange spill on the beach. This last spring, we saw them co-bloom with jellies. We had a kind of an influx of the water jellies here, and in the background you can see the egg yolk jelly. Egg yolk jellies eat other jellies, so the egg yolk jellies were likely following the water jellies here. And when you have a bloom like this where the plankton sits so high in the surface, while that bloom is doing really well, you'll have better visibility at depth. So if you're a diver and you see this at the surface, you're like, oh, okay, maybe I can see better. Just once I get down, it'll just be a little dark, bring a flashlight. But then once this bloom dies off, all of that organic material will start to sink in the water column, and you'll have a couple days of less good visibility down at the bottom. So it's really something to watch and kind of see how the pattern goes. Another thing we see here in the late spring to kind of midsummer is a conglomeration of harbor porpoises. Harbor porpoises are a marine mammal that lives throughout the Salish Sea. But we see them in really large groups here in the late spring to early summer. We think they might be gathering to feed and mate, but it is so exciting to see these little up to 5 feet long, so not super large marine mammals, just poke their little dorsal fins out of the water in these large groups. This background picture was taken off the end of the mast dock with my phone, so that's how close they were that my phone was not super blurry. This picture or this video was taken with my camera, so it was a little bit further away. And because these guys breach out of the water so short in duration, it only is like a second or two and they're back under. You want to look for, as Ariel calls it, the critter glitter, all of this reflection of the sun on their ripples to see where you can find them. A couple other things that you can see here during the summer. This is especially prominent in eelgrass habitats like we have here at the far north and far south ends of Redondo. Bay pipefish are kind of the Salish Sea seahorse. They're very closely related to seahorses, but they're kind of a stretched out version. Bay pipefish in this area will gather in the eelgrass beds or live in the eelgrass beds, but then they will often have their babies in the summertime. And like a seahorse, the males have a pouch that holds the eggs that are transferred from the female, and then they actually give birth to the babies when the time comes. And this little one here, I like to say they're about 1 inch long and about 3 hairs wide. They are very, very narrow fish, but they are pretty much fully developed when they are first born. They're just very, very small. Another fish that is constantly giving birth in the summertime are our shiner perch. Shiner perch are a perch species that reach maximum lengths of about 7 inches. So they're not super large perch, but they are live bearers. So the females will give birth to up to 30 babies that are about 1 inch long and fully developed when they are born each year. Here at the Mass Center, it's pretty regular that our resident shiner perch give birth in August. Throughout the Salish Sea, you'll see it from late May to early September that you'll see the females giving birth. So if you go to an eelgrass area or even around piers, you will likely see two size cohorts of shiner perch, the adults that are 2 to 5 inches long and the babies that are about 1 inch long during the summer. And then one other shout out. These are not my photos. These are from Mass annual photography contest, but they were such good photos that I wanted to give them a shout out. The blunt-nosed six-gill sharks, Hexanchus griseus is their scientific name. These sharks are large, usually deep water sharks that do tend to come fairly shallow into diveable depths here in Redondo and a few other sites, but Redondo has been as a very active dive site. There have been lots of sightings of six gills in the last, I don't know, decade or two. There's been some projects kind of studying why they're coming shallow and tons of great photography from the local divers, both in the Mast annual photography contest and other places like on Facebook. But these sharks are incredible. Usually we see the smaller end up to about 8 feet here in Redondo. And while they do come to diveable depths and sometimes even very shallow, like 20 to 40 feet, that's kind of an extreme shallow. Typically they are between 80 and 100 feet from about end of May to the beginning of September. So we're kind of wrapping up shark season, and they will come back again next year, hopefully. So that is the end of my Seasonality of Poverty Bay talk. I hope you enjoyed it. And this picture, I just love. I always have to end my slides with this picture. But yeah, if there's any questions, I am happy to answer them.
Awesome. Thank you. I've got a few questions, and if anybody has questions, please drop them in the chat box. But to, I guess, start off with, um, you were talking about that we've got a flow through system here. So would you say that the Mass Center Aquarium, when people come visit and see the animals in the tank, it's kind of a mirror or a lens to what's happening in the water at that time?
Yes, I would absolutely agree with that. Though we do have some animals that weren't originally collected right here in Redondo. The animals that we have collected here in Redondo definitely mimic their nature's counterparts here. And they also they're experiencing all the same temperatures and salinities and storms when the water is all mixed up and then it comes in through the pump. So yes, I would say that it's a good it's not a one-to-one comparison, but it's a very close comparison to what's going on in the greater Poverty Bay.
And with the flow through system, so people at home who go to aquariums all around Washington, we've got lots of them, but across the United States, closed system is, like a fishbowl at home, right? Same water circling around. Our water gets pulled in. We don't do any filtration here, but other aquariums do. So, say I go to another local aquarium, will I see the exact same stuff that's at the Mass Center, or would it be a bit different because they're not flow through systems?
So that's a great question. So the Mass Center is part of the Community Marine Centers of the Salish Sea, which is an organization of small aquarium facilities, kind of both in Washington and British Columbia. We've got some partners on both sides of the border. We are working together to kind of expand our knowledge and share resources. So I would say for the facilities that are showcasing native Washington species, you are going to see some of the same or organisms at each aquarium. You're probably going to see Shriner perch everywhere. You're going to see a lot of bay pipefish. But each aquarium kind of has its own focus. And so you're going to see special species in each facility that might be a little bit different. Some of our partners are open systems, just like the Mass Center, where they're pumping in water that's minimally filtered, just kind of keeping anything that might clog some pipes out. Or you have some facilities like Harbor Wild Watch in Gig Harbor that is a fully closed system, meaning they are either bringing in or making their seawater, and it's all self-contained. None of that is going back out into the Greater Bay. And then you've got your larger aquariums like Point Defiance, Seattle, and Vancouver, where they may or may not be pulling water from the Sound, but they have filtration and systems in place where they can keep both native species and then also tropical species in separate in the same facility but separate exhibits.
Awesome. So it sounds like there's a lot of challenges with an open flow system that knowing seasonality is beneficial, but also kind of like helps for when you know there's going to be things down the line happening, whether it's warm water or harmful algal blooms. So as a lead aquarist at an open flow system, what are some of those challenges, and how do you work around them, combat them, or what are some things that you just have to go with Mother Nature?
Yes. So it is so crucial to know what's going on in the greater ecosystem to best serve the aquarium. Because if I know that during the summertime we see higher temperatures, which puts our sea stars at a higher risk of wasting, I can either combat that by installing a chiller on a small tank so that we can keep that water colder, or just knowing the signs to look for so that we can preemptively treat with higher water flow or more aeration. Being able to kind of suspect that something might be happening, or if something does go wrong, looking at both what could have happened within the aquarium, but also was there a plankton bloom that came in the pump and caused issues in our animals, which can happen. And so, do we need to add some just very short-term filtration to our system, or do we need to do a specific, medical treatment? Knowing what might happen just gives us that many more options for responses if something does go wrong. That being said, we do have some positives to being an open system, which is we never know we know what's in the tanks, we know what we put in the exhibits, but the opportunity for new coxcombs in the touch pool, or new sea urchins in our surge tank, or new sea slugs in every exhibit. We get so much larval recruitment where the larvae come in as microscopic plankton and then settle into the tanks. It's really exciting, and it also shows us that these tanks are a healthy environment for these animals to settle into. So it shows that we're hopefully doing something right. So
So
definitely
our visitors
challenges.
need to Yeah,
Definitely
so our visitors
challenges.
need to come in like every quarter, basically, is what you're telling me. Is that like in
Yes.
order to see the seasonality, you can just come look in those tanks, but make sure it's, 4 times a year. Otherwise, you might miss some of the cool stuff.
Yes, I definitely recommend coming throughout the year, different seasons. Brave the rain in the winter. It's worth it, I promise. But also, yeah, just like seeing the seasonality. It took me about a year to really see that, even though I was familiar with the ecosystem and I was familiar with the animals in the Mass Center. Really seeing that those trends over the year, the first year that I was here, helped inform the second year that I was here. So it was really helpful.
You were talking about harmful algal blooms being one of those challenges. So before we get into the bad side of plankton or the sad side of it, what is the weirdest or coolest plankton that you've seen during the monitoring season?
I would say that the two species with eye spots are still probably at the top of my list for weirdest and also coolest. One of them I had known about from a previous mentor of mine who had that was his favorite species, or is his favorite species. And so I was like, okay, I know to look out for this, but I'd never seen it before I came to the Mass Center. And then we saw it like every week, and I was like, well, that's exciting. But the second one, the one with the piston, was one I had never seen and never really even heard of. So it was a really fun kind of dive into that kind of family of plankton, and then figuring out what genus and what classification it was in, was really exciting. And it's just bizarre to have a single-celled organism with an eye spot. So yeah, those two were definitely up there in my two favorites.
So even the tiny stuff is watching us when we're
Oh
we're
yeah,
in the
yeah.
main environment. So with those tides, you were talking about the algal blooms, harmful algal blooms. Some of them are fish irritating, fish killing. Some are harmful to humans. You're talking about Noctiluca here being totally fine, but in other places is harmful algal bloom. So one of the questions is, do we get red tides here in Poverty Bay?
Yes. So the term red tide is kind of a twofold thing. So yes, we get plankton blooms that turn the water a reddish kind of brownish color. And yes, we have blooms that can cause either human or marine life mortalities and sickness. But that's not always the same thing. So if we have a coloration change in the surface waters, especially if it's like an orange or a brown or red, it's definitely something to sample. And that's usually if I get here and I walk out on the dock and I'm like, I can't see more than an inch down in the water column and it's a bright, like brownish red, I'm immediately taking the plankton tow net out and taking a sample, looking to see what it is. But a lot of times it's not one of the toxic species. And then conversely, there can be days where the water is super clear and you take a plankton sample just because it's the day to sample, and you can find some of the harmful algal bloom species as well. So do red tides happen in Poverty Bay? Yes, but it's not always cause for concern. And it's really highlights the importance of keeping those partnerships with Sound Toxins and other organizations that do the monitoring, whether we're kind of the first line or if they're coming in and monitoring as well. Um, yeah, that's red tides do happen, but red tides aren't always the problem. So
So you talk about monitoring. How do people know whether or not it's safe to go into the water at those beaches? Because like that Noctiluca, bright orange, nature usually bright colors means stay away, but you and I go running straight for that water. How do people that don't know much about in the marine environment, where are they to get this information or know whether or not to touch the water?
Yeah, so I believe it's the Department of Health, but there's a map online you can search. Usually if you look for like shellfish fishing closures, those areas will the map will be updated online, whether it is open or closed for shellfishing. And if it's open for shellfishing, then it's been tested and there's nothing in the water that's going to hurt a person. Even most of our harmful species, um, there's no danger to the person touching the water. Um, it's if you eat something that has ingested that water, because it's the process of biomagnification, that clam or mussel has ingested all of the toxic plankton. It didn't hurt the clam or mussel, but it can hurt the person eating it because they have so much in the body. So generally, unless there's a sign posted on the beach, which can also happen if there's some sort of, sewage spill or something, the beach is safe to be on. But there are maps online. I believe the Department of Health is one of the suppliers of that. That you can check and just double check. You can also, if you're going like to Redondo, if there's something funky in the water, we're usually posting about it on social media. So we don't want anybody to be nervous going to the beach.
Awesome. So we are running out of time very quickly, but one last question. Talking about all of the babies that happen here in the aquarium and larval rearing. So, what are some of those challenges, and what are some of those successes? I know you talked about the German croaker paper that you wrote, and our babies being transferred to other aquariums to share with them. So, yeah, what's the challenges? But what are some of those really cool things that you found out during that process?
Yeah, so there were definitely both in all of those experiences. Challenge is definitely raising fish in a minimally filtered system. With the larval fish specifically, we did filter the water that they were getting to make sure that it was fairly clean for them. But you can never filter something 100%. And so dealing with parasites that come in the water was definitely a setback to some of our larval rearing adventures. But the payoff of being able to see for the grunt sculpin that whole development through the plankton stage was really fulfilling for me. And then for the lump suckers, just seeing them go off into the world was like, they're my babies. They're going off, and it was like babies going off to college. It was very exciting to see kind of that whole process from start to finish. So yeah.
Awesome. Well, thank you for this amazing talk. For those of you who are watching, if you don't want to bear the cold and sometimes murky water, as Emily was talking about, but you want to see the really cool seasonality shifts and animals that come and go, check us out at the Mass Center Aquarium here in Des Moines on Redondo Beach. We are open Saturdays from 10 to 2. You can also check out some of the past talks that Emily has done. She kind of went really quickly through all this cool stuff. We have more in-depth talks on our Science on the Sound Speaker Series playlist on our YouTube channel. Also, if you subscribe to our YouTube channel, you will get updated on when we go live for our other speaker series and talks that we do. But thank you, Emily, and good luck on your next adventure. We hope that we see you again soon. And maybe when you're done with your master's, we get a thesis talk in 2 years.